People, why don't you return the homework that I forced to give back? You have your, or you, no.
You want this week's homework?
Okay. Okay. So what I'm going to do is give an overview of fractals. So if you've had my other course, you're going to get it again. But it's going to be slightly different. At least I redid the order of some of the transparencies. But it's basically the same thing. And as I mentioned probably a number of times, it takes me at least 3 times before I get something anyhow. So I'm going to run through it again. And maybe it will be useful having it more than once. And I'm going to try and talk slower. I give this is a version of the overview. If you remember from the other course, I give one overview lecture and go back and do things in more detail. But usually I give that lecture talking very fast and show too many transparencies. So now I'm going to show even more transparencies, but I'm going to talk a lot slower and spend more time on each one. So I get the slide over here, and to push this button, which I've already pushed. So if you could put on the, yeah, thanks. And does the pointer work on this? Yeah. Let's start off with the difference. What I've done in this presentation, and this is a lot of stuff from my book also. So this is actually the order in which things are presented in the book. So you can picture me holding up the book at this point and plugging the book. And the book is called, I don't have to say what it's called because if you log on to my website, you can get a URL directly to the publisher and order it. So I don't even need to say what it is because this is the end of the 20th century, so I just have to tell you my website, which you all know. So the way I've set this up is to always try to compare a fractal and a non-fractal property so you can see how one compares to the other. So if we have a non-fractal object, like this, this circle, and we enlarge it, it gets bigger because we're looking at it in finer detail, but nothing changes in it. If it was smooth to start off with, the radius of curvature means that it looks smoother, or we see less of the curvature. But it doesn't look like there's any change in the nature of the object itself. And this is the way we're used to thinking about a lot of objects. But not everything has this property. So for example, if we had something that might be the lining of the intestine, if we enlarge it, you would think that this area which looked sort of smooth will look even smoother, because that's the situation we saw here. Here we have something that looks a little bit curved. When we looked at higher magnification, it looks less curved because we're seeing a small piece of it. But in terms of the intestine, when we look at something that looks a little bit curved, when we enlarge it, it doesn't look smoother. In this case, as we go from the intestine, we begin to see villi on the intestine. So we have properties at the next finer scale that include more details and more information than what we saw at the previous scale of lower resolution. Now, one of the things about this is that what we see at this higher resolution is not an exact copy of what we see at the lower resolution, but it has some relationship to it. That is, if this consists of hills and valleys, then at higher resolution, we also have things that consist of hills and valleys. So there is some sense where the properties at this higher level of resolution are like the properties at the lower level of resolution. That is, this structure is similar to itself at different scales of magnification. And that's what we're going to call self-similarity. And in this case, in the intestine, as we continue to magnify it and get down to the cellular level, so if these represent now individual cells, we'll see microvilli, projections on the top of each cell. And again, these microvilli are not exact copies of the villi or of the intestine, but they have some properties that are similar in the sense that we have hills and valleys. So this is one of these essential ideas of fractals, that we have structures that are similar to themselves at different magnifications. Now this is kind of a new sort of symmetry. One way to think of this is that, maybe if we switch to the Elmo for a second, we're used to symmetries as I may have said, I usually spell things like symmetry with all these double letters because it makes it more symmetric, but I'm not even going to try to spell it right this time. We're used to symmetries like translational symmetry, which means if I transport something from here to there, it looks the same, for example, like a line. Or rotational symmetry, where if I have an object like this, we really think that's the same object as this. And the idea is that things are invariant under these transformations, these symmetry transformations. But now the transformation we're talking about is a very different one. It's a self-similarity transformation. That somehow an object is the same when I take smaller and smaller pieces of it. And so this is a different sort of symmetry. But in some ways, this is an extension or is related to these other types of symmetries as well. But it's a new type of symmetry. And in fact, many of the properties of these self-similarity transformations can be written in terms of translations and rotations and things like that. In order to do it, we would use not only translation and rotations, but something which I'll call a contraction. A more formal definition might be called an affine transformation. And an affine transformation is something, for example, that makes a big thing smaller, which is why I've called it a contraction. And now you can see that or the fractals that we'll see, by translating and rotating and making things smaller, we can take the whole object and reproduce smaller copies of it. In terms of these mathematical objects, the smaller copies are exact copies. In terms of this more biological fractal that might represent the intestine, the copies are not exact copies, but they are kind of approximate copies. Now, another way of describing this is looking at the sizes of the features of something. Or, as I said before, these could represent spheres in space. I'm saying spheres instead of little virus particles. Or they could represent symbolically the values of numbers of things. And I've drawn them here, or told someone else to draw them, all of the same size. It might have been better, and I have in some later transparencies, drawn these not at the same size, but have most of them roughly the same size and some bigger and some smaller, forming perhaps a Gaussian distribution. But the basic point here is either for this object or for this set of values symbolized by this object, there's only one characteristic scale, which is about this size, that all of these are about the same size. And so if I had a ruler that was finer than this scale, I would measure all of the properties of this set of object or these set of values. That having finer resolution doesn't show me anything more about these things. But that's not the case for a fractal. So this shows what I was showing at the end before we took a break of an object or a set of pieces that has a few big things, lots of medium-sized things, and a huge number of small things. And now, and as I said, we can think of these either as pieces of an object, or we can think of these symbolically of values measured from the experiment. So this would mean that maybe we do the experiment sometimes and see a big value come out of the experiment. Most of the time we see middle-sized values come out of the experiment, and most often we see really small values coming out of the experiment. So now we have not one single characteristic scale, but we have features that vary over many ranges of sizes. And in this case, these are some of the sizes of the features that are present in this object. Now, these fractals have a number of different characteristics, and I'll first describe 4 different properties the fractals have, and then we'll discuss those in much more, in somewhat greater detail for each property. The first one is this symmetry relationship, which I'm calling self-similarity. And I've illustrated that here with a map, a symbolic map of a coastline. So this is a coastline. It doesn't look like South Florida, but maybe it could be. It feels to me like Fort Pierce. But it doesn't matter. Although the water isn't blue, at least the land is darker than the water. So we have a coastline. And somehow you would think that if you enlarge the coastline, it looks wiggly. You would think, and this is what you're normally told, that when you get a bigger magnification of something, if it looks wiggly, you should resolve it and it should look smooth. But in fact, our experience with real objects is that that's not necessarily the case. That we enlarge a piece of the coastline, and rather than looking smooth, it looks just as rough. This enlargement of the coastline of this little piece looks just as rough as the whole piece. And again, if we continue to enlarge a piece of this coastline, it looks just as rough as the whole thing. So the coastline, the perimeter of the coastline, the place where the water meets the land, is as rough as at different scales. And it is similar to itself at different magnifications. So we have this symmetry of self-similarity. Now, why this should happen is really kind of not so clear. We're going to see that in some fractals we get fractal behavior because we have an underlying physical phenomena that then boots itself up across different scales. In fact, if you could switch to the camera just for a moment on me. Thanks. Thanks. We're going to see that we can get self-similarity because we have structures at one scale that somehow bootstrap their way up to other scales. It's not the world's best graphic, but I felt I needed to do that. So, in fact, what Laura would have talked about today in the Journal Club is how properties at the molecular level, which we would think of normally in terms of milliseconds, can actually bootstrap themselves up to things that can last seconds and minutes, and therefore may affect spike trains in cognitive processing. So in that case, we have something at one level of organization that is able to reach to much larger levels of organization. So that's one way we can get a fractal. But another way, if you go back to the computer, thanks, is in this case, maybe that's not what's happening. It's somehow what sculpts the coastline at the scale at which you could put the sand on your bucket and make a little bay and peninsula is maybe different from waves that work on a scale of a half mile that are moving sand from one jetty to another jetty. And so in terms of the coastline, it may not be that what's going on at this scale is the same process that's going on at that scale. So it's possible that in a situation like this, what we have is we have different processes at different scales, and then somehow mystically, in a way that we don't understand, these processes communicate with each other to create a consistent pattern so that the wiggliness of one scale is reflected in an equivalent wiggliness measured at other scales. This issue of how we get things across scales is not a clear issue in a lot of different ways. So where the self-similarity comes from, whether it's one process booting itself up in terms of the inactivation of the ion channels, a similar thing happens in terms of magnetism. If people were at the talk on Wednesday, for example, where he was discussing ferromagnetism. In ferromagnets, we have domains on the size of microns that get organized to form domains on the size of centimeters. And so we have one process that bootstraps its way up in scale. So that happens, but we may have different situations where the fractalness arises in a different way of things talking to each other. And what could it be, the information that they're sharing to form a consistent pattern is not at all clear. So that's an unresolved issue. Second property of fractals is scaling. And what I mean by scaling is that if the coastline has wiggles at different length scales, as we measure the length, we'll find different values for the length when we measure it at different resolution. So we're used to thinking that there's one value for something. In fact, in terms of these features, we would say there's one average value for these features. But in this case, there's no average value. The value we get for the size of these things depends on the resolution at which we measure them. And I'm going to call that scaling. So this is very different now from handling experimental data in the way we're used to handling. Because now not what is important is not a single value, but how that value depends on the resolution at which it's measured. So we're dealing in a different world now where we have to have the values, we have to have more information. It's not good enough to have one value. We have to have enough measurements to get different values in order to tell what's going on. And we'll see that there's a parameter, a value that characterizes the scaling, and that parameter that tells us how these values depend on the resolution, that gives us a quantitative measure of self-similarity, is something called the dimension. And I haven't said here what the dimension is. I've tried to illustrate it symbolically, where if we have a line segment viewed at low resolution, then if we view it at higher resolution, we may see that this one line segment consists not only of just one piece, but 1, 2, 3, 4 pieces. And so the dimension we'll see quantifies, as we go from low resolution to a higher resolution, how many new pieces appear. So the dimension is really giving us a quantitative valuation of the self-similarity, how many new pieces appear, a quantitative valuation of the scaling. And lastly, which is the reason why I'm introducing this in this course, is that we have these statistical properties, which, oh, that's weird. We have different colors on the different screens, actually. Oh, that's interesting. So I better look at the screen to see what colors you're seeing. On my screen, this is red, even though it's blue on your screen. So that's a big difference from red to blue. I don't think this changes anything. So I don't know why that should happen. And on my screen, these are blue, and they're black on your screen. All right, so I have to watch screens. And this overlay has to do with a font problem. Basically, I've already said something about the statistical properties, that we have a situation where moments of the distribution may either tend towards 0 or not a finite limiting value. So we may have examples where the mean goes to 0 or infinity, where the variance goes to infinity. And this is the main reason why I'm talking about fractals in this class, and also I think the importance that fractals will have in analyzing data in the future. That we're all used to dealing with these distributions with finite moments. And a lot of the real data in the real world does not have these properties. And the fractals allow us to create a mathematical analysis that's more consistent with the real properties that the real data has. And that's what I think the real utility of the fractals will be. So let me discuss now each of these properties in a little bit more detail. So first of all, let me talk about self-similarity. There are at least two major types of self-similarity, which I've already hinted at. The first one we'll call geometrical self-similarity, or mathematical self-similarity, where the magnified piece of an object is an exact copy of the whole object. So for example, if I take this object, and this is called the Sierpinski triangle. And let me give you a hint of how it's constructed. I start with a solid black triangle, but I take out one of these triangles. And now you can see instead of one solid black triangle, I would have 3 solid black triangles. But then from each of those 3 black triangles, I also take out the middle triangle. So I then take these 3 out. And then that leaves 3 smaller little black triangles within each of the small ones. And then I take out another triangle out of that, and I keep going. If you think about it, eventually I take out enough of these so there's no area in this left at all. It just points, no area. So the area of this thing goes to 0. And this procedure then results, so if I take one piece of this and expand it, since I can keep going finer and finer taking these pieces out, each piece of this thing looks like the whole thing, exactly. And that's called geometric self-similarity. Now, one of the problems, or it's interesting that when we present almost any subject area, how we present it conveys some sense of what it is that's being presented. And sometimes we take things out of historical context so that it looks like it's something, even though the underlying reason for something might be very different. And what all that rather ambiguous language means is that there's a sense that comes out of fractals that fractals were developed to handle images or arise out of handling images. And in fact, the ability to handle images is very important. It makes the fractals very clear. But a lot of the fractals arise out of mathematics and do not arise out of analyzing images. And in fact, the reason why this object exists in the first place has to do with the solution to some rather abstract mathematical problems. In this case, either this or the Sierpinski gasket, which looks similar to this, actually arises from asking the question, how complex a line can you draw in a space? And this figure is the answer as a proof of a theorem to that sort of mathematical question. It's not exactly the right question because I can't remember what the right question is, but Hans Otto Peichen knows what the right question is, and if I had my notes from a lecture that he gave in front of me, I could tell you what the right question is. But the point I'm trying to make out of this, as he makes, is that we're so used to seeing the graphics of this that in fact these objects have been around for about 80 years and people never saw the graphics. So we're in a tremendous situation of having the computers make it possible for us to see these objects so we can get a much better feeling and learn a lot more about them. But they're actually not pretty graphics. People are down on fractals sometimes because they think it's just pretty graphics. But actually, these represent a lot of very deep underlying mathematics. And these figures are solutions to theorems that describe some mathematical properties of the world. And it's interesting that now we think of them more as graphics because we can see them. The people who did these theorems actually never saw these objects or only had a vague hint of seeing them because they didn't have the computers to draw them. So one of the points I'm trying to make here is that there's a lot of underlying real mathematics. And these things have meaning as well as being objects. But usually in terms of exactly geometric self-similar objects here, this is rarely what we have in biology. We rarely have things that are exact copies. And I showed that in the first example of the fractal I gave in the intestine, where we had villi, we had undulations in the intestine and villi and microvilli. They're not exact copies of each other. We're in a different situation, which I've called statistical self-similarity. And this illustrates how the crypts in the intestine, so I managed to think of the word after treading water for 5 minutes on it, how the crypts in the intestine are related to the villi in the intestine. So there are, these shapes are kind of like these shapes. They have hills and valleys, but they're not exactly alike. So the way we relate this not exactly alike is through a property called statistical self-similarity. And what that means is that some measure of this is self-similar to this. And one of those measures could be the average length along the perimeter. So the average length here is similar or self-similar to the average length here. What that means in terms of scaling is that the length measured at one resolution is proportional to the length measured at a different resolution. That the length measured at a finer resolution is going to be much longer because it includes many more of these undulations, and so as we measure it, we pick up a larger and larger length. And so this means that the length here is proportional to the length there. And it means it's proportional. If we go down a factor and a half and see the length double, if we go down another factor of a half in resolution, the length will change by another factor of 2, etc. So there's a proportionality here. So the way the length changes depends on the proportion. This can also be described in this equation, which you may or may not be able to read at the bottom here, which says that the probability density function, which is the number of pieces of a given length. Oops. So let's see if we can get this going again. Which means that the number of, let's see now if I'm smart enough to watch this say stupid things like it's checking the memory and speak at the same time, which I think I can do, but only if I don't look at it. The probability density function measured at one resolution is geometrically similar to the probability density function measured at another resolution. So it means that the distribution, the relative distribution of large and small pieces at one resolution is similar to the distribution of large and small pieces at another resolution. That's what dimension means. That if we go at a big scale and go half a scale, we see 3 new pieces. And in a small scale, if we go to another half scale, we also see 3 new pieces. So so we can express this mathematically more formally by saying the probability density function measured at one resolution is similar to the probability density function measured at another resolution. So we're doing okay so far here. Okay. I can do 2 things at a time, but not 3 things at a time. So I can talk and push the button, but I can't talk, push the button, and watch the screen. But in general, that's true for me. I can do 2, but not 3 things. So if I'm listening to music and reading a book and eating a sandwich, either stuff in the sandwich drips on the book, or I eat the sandwich, but I lose the line that I'm reading in the book. So in general, I can do like 2.5 things. I can do 2 things okay, but I can't do 3 things. So looking at the screen was one too many things, so I had to stop talking. But now I can talk because I'm not looking at the screen. So again, we have this concept. Let me draw a picture of this while that's booting up anyhow. So what we're saying is that the probability density function measured at one scale, for example, millimeters, will have a certain shape. And the probability density function measured at centimeters will have some shape. And these 2 will have a similar shape. And the way we can express this is to say that the probability density function of some property measured at resolution A of R is similar in distribution, which is what this symbol means, has a similar shape to the probability density distribution measured at Q of R. And this is the formal definition of statistical self-similarity. And you can see what this means. It means that the number of pieces, so it means that if we have big pieces, they have smaller pieces, and smaller pieces have even smaller pieces. But the distribution that says how we go from here to there is the same distribution that we go from here to there. And the same meaning that there's a geometric self-similarity that links these 2. So that's why this is called self-similarity. And it's, again, like a symmetry, like a scaling. In this case, it's a contraction on how things at one spatial scale are related to another. So this is the formal definition of statistical self-similarity. Can we catch up here? Okay. And let me show you some examples of this. And I'd like to show you examples. Since fractals are always emphasized as objects, I kind of want to not do that. I want to emphasize to you that I'm thinking of fractals in terms of these statistical distributions, in terms of stable distributions. So when I say fractals, I'll always try to say either patterns in space or properties in time, or values of sets of numbers. So maybe that's worth writing out explicitly what I mean by fractals. So what I mean by fractal is not just an object. So a fractal is not just an object, but it could be an object. So fractals include objects. They include things that have this probability density functions, which I've called stable distributions, and they include sets of numbers, and they include processes in time. And again, what I'm trying to do with this, I'm using the word fractal. I'm extending the use of the word a little bit. Although what I'm defining fractal is consistent with formal mathematical definitions of fractals. So I'm not cheating here. But I'm more using to describe what would really be, in terms of probability density functions, just be called stable distributions. I'm trying to give it, I think, a name that represents more of the properties of that. And I think fractal is an appropriate name. and as I said, even a mathematically correct name to represent that. So we're not just talking about objects. And that's why I'm talking about in this class, because many things in the world have fractal properties and have statistical properties that cannot be adequately addressed, characterized, described, interpret, represent data, and lead to models of data in terms of the Gaussian statistics that we've had before. So if I get back over here, let me now give you examples, as I said, both in space and in time, of fractal patterns. And the first fractal pattern I've put down here are nerve cells. Now, one of the things about these fractal patterns is that these are nerve cells growing in a rather flat culture dish. So we're really talking about two-dimensional patterns. If these were three-dimensional, like that, then we would see projection effects like this. Thank you. You can switch back now. Thanks. Well, it wasn't up on the machine before. People would have no idea what I'm talking about. They would just hear you laughing, and they went down. If we would have something three-dimensional like we saw briefly before, then we would have projection effects. We don't have that here. These cells are lying flat. The retina is quite flat, and they're lying flat on the dish, so we have these cells. A number of people have been analyzing the fractal properties of nerve cells, including people in Boston, Caserta, Stanley, Eldred, Decord. Most of these people are in Boston. I see this guy next Friday, actually. And a different group of people, Tim Smith, forget Smith's first name, in National Institute of Health in Bethesda, Maryland. And they've been looking at fractal patterns. What this group in Boston has been doing is there's a mathematical mechanism that would generate patterns like this. And they're trying to see that mechanism involves properties of diffusion. And so they tried to see if they changed the medium in which the cells grew, and therefore changed the diffusion properties, whether it would change the structure of the cells, which it didn't. And the group headed by Smith has been looking at using this dimension to characterize the shapes of different types of neurons, different nerve cells, to see if they can use this as a way of characterizing or describing the nerve cells. And a group that I collaborate with in Poland and in the United Kingdom has been using fractal dimensions to measure nerve cells in the retina and correlate that with electrophysiological properties of the nerve cells. And they found some weak but some correlation between the shape of the nerve cells and how the nerve cells fire in time. So they're trying to use this as a way of characterizing. Specifically, they're looking at amacrine cells in the retina. So here are nice patterns. Let me not lose it so fast. And again, why these are self-similar is because we have these little branches here which are reflected or self-similar to bigger branches, which are self-similar to even bigger branches. So what's self-similar here is we have branches which have branches which have branches. Another example is shown here. These are, on the left, blood vessels in the retina measured by Barry Masters, who's in Washington, D.C., and Faridun family, who's in Atlanta at Emory. Mainster is in the Midwest, I don't remember where. And what both of these groups found, this is actually, although I made these, they're blue on your thing, they're red on mine. I made them red because, even though you see them as blue, because to symbolize blood vessels. But when this measurement is really done, they're actually kind of light yellowish green. Because what was done here, no, it's good. What was done here as a dye fluorescein was injected into someone's arm. And what's nice about eyes is that they really, I don't know if they're a mirror to the soul, but they're a mirror to what's going on inside the body. Because it's one of the few places you can see capillaries. And so looking into the eye and allowing you to see blood vessels in the eye, even to small capillaries, provides very good diagnostic information on a lot of disease processes that are changing the capillaries and changing the blood supply. So it's very diagnostically useful for diabetes and a number of other conditions. And so in this technique, this dye fluorescein is injected into the body. And then ultraviolet or blue light is shown into it and fluoresces in this yellowish green. Now, the whole person doesn't fluoresce yellowish green, so it's not like X-Files or something. But the only place that fluoresces is these blood vessels in the eye because they're superficial. They're in a place that you can actually see. So this is a way of measuring what's going on in the retina. If these blood vessels leak and the fluorescein spills into the retina, you can see that there are problems, and this happens in diabetes, for example. Depending how different areas of the retina light up, tell you lots of interesting things. If we go back to the computer, you can see this is the place where the blood vessels enter into the eye, and they snake around different parts of the eye in the retina. You can see they're avoiding one place over here, which is the fovea, which is the highest density of photoreceptors, and so it stays out of the way of light hitting them so you can see a sharp image. And this is a typical pattern. So this is the fovea. Only about 2 degrees of your field of, if you go back to the camera for a moment, only about 2 degrees of your field of vision is in clear view. You want to punch up my hand again? Why don't you go back to the camera? Only about 2 degrees of the field of view is in clear focus, which the full moon is about half a degree. So it's only about 4 times the size of the full moon is in focus. Everything else is fuzzy. And what you do is you take that clear thing and move it around things that look interesting, usually edges or flashing lights or things you're interested in. And that creates an image that has much finer detail, but most of it is fuzzy. And so your brain back here somewhere makes this idea that things are really nice and sharp and in focus and level and everything. But that's just a fiction that's made up by the fovea. So if we go back to the computer again, you can see that it's only this very small area that's actually in sharp focus. And so we have these blood vessels that are voiding that area, and they have this fractal pattern. So we have little branches, big branches, and much bigger branches. So we have this self-similar fractal pattern. And we have a similar condition now in the airways and the lungs. Now the retina is pretty flat, so this is again pretty much of a 2-dimensional image. This is a more complicated image because we're viewing things 3-dimensionally. And so we've compressed them, shadowed them onto a 2-dimensional plane. So this is a little more complicated. And what we're looking at here are the bronchial passages, the passages that bring air down through tubes in the lung into different pieces of the lung. And again, in this case, we have almost 12 generations of branching. So we have, you can see a big branch up here and finer branches down here and finer branches within the lung. So we have self-similarity here in terms of the branching. Well, those are branchings or self-similarity in space. I wanted to emphasize self-similarity in time. These are some measurements done by Gaeva and Kiza. In fact, I see this person next week also and got email from him today. All these people are physicists and will be at the American Physical Society meeting next week in Los Angeles. This guy is sort of interesting because he was at a Biophysical Society meeting actually presenting the work I'm going to describe here, and no one seemed to pay much attention to it. He had a poster. No one seemed to think it was a big deal. But I talked to this guy at the poster, and he seemed particularly clever at explaining things. He just seemed sort of sharp. And no one was interested in this because this was fractals, and people in Biophysical Society don't think much of fractals. But I found out actually a few months later that what's interesting about this guy is he has a Nobel Prize in physics for doing something in solid-state physics. But since he was at the Biophysical Society, even though these people say they're biophysicists, none of these people know anything about physics, or very little, and so they didn't know that this guy actually had a Nobel Prize in physics. And so no one was speaking to him at his poster. But it was interesting. This guy just seemed really a little bit brighter than the average person you speak to at the poster, and just a little bit sharper. And what? Well, I don't know. Not always. I met Josephson recently at a meeting in New Hampshire, and he's pretty smart. The guy from Josephson Junction, on which SQUID semiconductor quantum interference devices are based that measure magnetic fields in brain, among other things. And he's into like sort of trans-sensory perception and all these other sort of odd things. So he's smart, but sort of little further out. But I shouldn't really quite say it now. I'm not really trying to disparage what he was doing. I think there were interesting issues he was working on. But to get back to Giever here and his measurements, they developed a very clever system where they have cells growing in culture on a substrate. And when you look at these cells growing on culture, they don't look like they're doing much. They're sort of sitting there, looking at a microscope, put fluid on them. They're sort of, sometimes detach and move around. But these cells are really living things, and they're actually moving, right? That's what living things do. They move, right, and use energy and stuff. And it turns out these cells are forming attachments to the substrate, and the number of attachments varies in time, and they sort of squat down and sort of lift up attachments. So the average height, or the height averaged of these cells over the substrate, changes in time. And what he did is to develop an electrical circuit based on an oscillating current sent in and a lock-in amplifier and a data acquisition to measure the height of these cells in time above this substrate. Now, at a meeting I was at actually last year in Poland, someone from Scotland was telling me that there may be some problems with this system, and whether these measurements are real or represent an artifact of the system wasn't so clear to him. And he said that had been published, a critique of this article from which this was taken on in Physica D had been published, but I haven't seen that. So I can't comment on whether this system is really working or not working. But let me show you some of the data that comes out of the system. So these are variations in time of the height of these cells above the substrate. And here, when we look at very long time intervals, we see fluctuations, big fluctuations in time. But when we look at a smaller interval in time, we still see self-similar fluctuations. And at a smaller interval in time, you can see these numbers are getting smaller fluctuations in time. And again, at smaller time scales, even smaller fluctuations. So we have a self-similar fractal type fluctuations of times in the heights of the cell surfaces versus time. And another example is shown by measurements that I've participated in. This is from a group across the street from NIH at the Naval Medical Research Institute, involving a number of people, including Steve Yandel. Yandel, who retired very recently, was actually the first person to measure quantal electrical signals from light impulses in the horseshoe crab limulus. And so was one of the people who pioneered measurements of electrical recordings from photoreceptors. And what we found here is that as we measured the voltage, in this case using the whole cell patch technique from T lymphocytes, that it's fractal. There are little fluctuations in time, and bigger fluctuations in time, and bigger fluctuations in time. So we have self-similar fluctuations in terms of the voltage that's being recorded across the cells. I should say, in general, people have always assumed, as a first guess for something, that, let me do this on the Elmo, basically, because I think this is an important point. It's a point I make in the other class, and let me make it here, that people always assume that the first guess for something was that it was smooth, or it had a single value for a measurement. And what we're replacing this by is a concept here in fractals that our first guess is something that things are rough, and that there is no single value, and that we have features at all scales. Okay. So, and in a way, these set of assumptions that really come out of this more fractal viewpoint, in a way, are much more like the real world. The real physical world, the real biological world, in that, in a way, the real chemical world.
What do you mean by no single value?
We can't characterize let me take a chemical example. Let's say we had something that had a lot of holes in it that was like a solid catalyst. We couldn't characterize the size of the voids in the catalyst by a single number, that there are big holes and smaller holes. And as I said, formally, we will see that for these distributions, the means don't exist. So there's no single value like a 0.5 centimeter that characterizes the voids here. That is, the voids will have this property like the Sierpinski triangle, which I can illustrate in a different way here, that there are ever smaller and smaller voids if each of these is a void in the
So you would be able to determine the surface area with a single value?
Right, right. We couldn't determine the surface area or the volume or the length if we were just looking at the shadow of the edge with a single value.
You could have like a fractal dimension, and that is single.
Right. Well, the fractal dimension tells us how the number of pieces or the area changes with size, but it doesn't tell us the size of something. So we switch to a different paradigm. In fact, that's worth saying here. The first guess before would be to ask a value, let's say, of the size of something. And now instead of asking, so we cross this out, instead of asking what's the size of something, we ask how the size depends on the resolution. Okay, so we've changed the paradigm between how we're looking at things. Let me also say that I think these lessons which I'm giving to you here, or even though they sort of look transparent or look very obvious, I think it's very hard to realize what the real lessons here are. I think it's been hard for me to realize. I think it's been harder for other people to realize. And we can write the mathematics of this in a relatively clean way. But to really see what this means in terms of how we think of things, I think has been a difficult lesson to really sink in. I think it's been difficult for me. And what I'm presenting to you, the aspect of dealing with the fractals in terms of the statistics, is basically something that I've been pushing. I think that the other people who deal with fractals like it and have been saying it, but have been saying it in an implicit way and not as explicitly as I've been saying it. And I think they like having it said explicitly too. So it's been a good thing to do. So I think these ideas are really difficult. I think, to say that suddenly, doing one measurement or one size isn't enough when that's what we're always used to doing in science, to say that we have to handle things in a different way, that we have things across scales, and how are we going to do all these measurements? And if we don't have a single value to characterize the voltage of a cell or the interspike interval, and how we're going to handle things, it's really a very, very big change in how we're doing things here. So this is really a revolution. And again, as I'm going to develop over the next couple of lectures, we're going to see that these statistic properties of the fractals are radically enough different that we can't define a mean. And that's such an obvious thing that you cover it real fast in the statistics course that you take. You have a definition of mean, you add them up and you divide by the number, or you integrate the probability density function weighted by the values. And so we have very different, very different, much broader thing going on here. Now, when the fractal dimension is an integer or a 0, we recover the case that we had before in terms of the Gaussian statistics. So again, the fractals are not something different, but are more general than the cases we've had before. Okay. So let me get back to here. And this shows another example in time of a fractal. And this is an example of the sort of thing that I've been working with. And these are currents measured through ion channel, electrical currents. This was actually done by a group in Washington University at St. Louis. And what they did here, this is a plot of current, I should say current versus time. And in this case, when the channel opens, when the current increases, this trace moves downward. And these are cells which are islet cells, beta cells in the pancreas. These are cells that respond to changes in ATP level and respond by producing insulin. What happens here is they've made a recording that they've played back here at relatively low time resolution, but they can play back this recording at higher time resolution. So in this case, there's one opening of the channel. You can see this is 5 seconds and 5 milliseconds. When they play one opening of this channel back at a higher time resolution, that one opening really consists of lots of opening and closing. So it's really self-similar. But it's not exactly self-similar. It's not as if this trace is exactly the same as that trace, but it's statistically self-similar. The statistical properties of how we switch between the opening and closings, I think you can see even here in a qualitative way, seem similar to what's going on there. And in order, we can quantify this. If this is statistically self-similar, what I told you statistical self-similarity means is that the probability density function computed at one time scale is similar in shape to the probability density function computed at a different time scale. And we can see that that's actually the case. So this is the probability density function. And how this was computed at different time scales was I took the data. In this case, it was me. And this is in the old days before I tried to be an administrator and run things when I actually really did stuff. And we had a tape recorder that I built the modulation-demodulation circuit for that we recorded. So it was an FM modulation-demodulation circuit that lived on a one of these boards where you put the wires in. It's not a breadboard, but the little white things where you put the wires in, so they shook loose all the time. You had to, it was an old 1950s tape, reel-to-reel, like 10-inch tape recorder. In any event, what we did is we had this data stored on the PDP-1134 computer, played it back into the tape recorder, or I had actually the data on the tape played into the computer. And we could play the data in at different analog to digital sampling rates. So that determined the effective time scale at which we're analyzing the data. And then what I've done is I've done a frequency histogram, just the way you did the histogram in the very first problem in this course. And so I have a histogram of how many times I have, in this case, closed times of the channel between 0 and 20 milliseconds, 20 and 40 milliseconds, et cetera. So this is the closed time, and this is the number of times in the bin. This is a semi-log plot because you can see this distribution looks at early times like a single exponential, but it's not a single exponential. There's always a tail sticking off here. And now, so this is the probability density function, and we can see how this probability density function looks at different time scales. So this is when I sample the data at 5,000 hertz. This is now when I sampled the data a little slower at 1,700 hertz, and it has the same shape. Now the numbers here are different. If you look at these numbers compared to the previous numbers, this is now smaller, this is faster. When we sample fast, we catch the short close times. We sample slower, we catch longer close times. You'll see on the subsequent ones these numbers will get even bigger. But it's the same shape. It's sort of like a single exponential with a tail. We sample at 500 hertz, the numbers are bigger. Again, a single exponential in the tail. And 700 hertz, a single exponential in the tail with even bigger numbers. So this means when we sample very fast, we catch the fast closings. But the distribution of those fast closings, which is here, has the same shape pretty much as the distribution of the slow closings. So this is an example of self-similarity in time, where we see that we really have geometric self-similarity in terms of the probability density function, just what I said. So this raises a question about what's going on in how the body is formed and how genes are expressed to determine shape in the body. In some ways, what's hard to understand is why we only have 10 to the 5th genes, 100,000 in the DNA in the human body. But we can form structures like the heart, which has 100 million capillaries, or the brain, which has 100 billion nerve cells. As I'm fond to point out, there are as many nerve cells in the brain as there are stars in the galaxy, which is about 10 to the 11th. And so this is a pretty complicated thing. But how can we generate something with all this complexity if the number of units that generate it is small? And one possible answer to this is that what's going on in the body is that the genes act through a repeated set of rules. You've seen in terms of the Sierpinski triangle, in fact, let me switch back to that again, that one way to make a fractal is to have repeated rules or iterated rules. So let me make a different fractal. This is from, I think her name is Helga, but it's von Koch. There's a, I think, Swiss mathematician of the last century. And she proposed this curve, which is called a snowflake. And the way we construct this curve is we start here with a triangle, and then on each side we divide the sides into 3 and add another little triangle that's self-similar to the original triangle. So each of these little triangles is kind of like a smaller copy of this whole big triangle. And we can keep doing it. So if I do it on this piece, I add another little triangle, and I keep doing this. Now, look what happens. Every time I have a triangle and add another little triangle, if this was 1 unit long on a side, or if this was 3 units long on a side, from here to there, 1, 2, 3, now it's become 1, 2, 3, 4 units. So at each stage in the construction, each side goes from 3 to 4 units. So if this initial triangle was 3, 3, and 3 was 9 units big, at the next stage of the construction it would be 4/3 times that, and at the next stage the perimeter would be 4/3 long, etc. So if I keep doing this forever, the perimeter of this thing is infinitely long. Okay? And we'll see that the fractal dimension of this is going to be related to how many new pieces we see as we change the magnification. But the point is we have these iterated, repeated rules in generating the fractal. This is a little bit different than what we're used to in terms of functions in mathematics. For a while, we're used to functions that specify one rule. So given x, it tells you y. But here it's not like that. We have to repeat things and repeat them, and from that generate the function. So this has a little bit of a different flavor. We find things through the constructions of things, and so it's a little bit different. Okay. And that's what may be the case in how things in the body are formed. That is, we may be in a case in the body where we have the genes, and then repeated applications of these rules of the genes are then formulating these complex structures. I would say this is a little bit controversial, but it's an interesting way to think about, and an interesting way to try to understand how a limited number of genes can form a much more complex set of structures. So let me now describe a little bit more in detail about self-similarity. The self-similarity means that we have smaller and smaller pieces. And as I said, as we add all those pieces together, for example, in measuring the length of the coastline, that self-similarity is the reason why there's a scaling. Because as we look at the coastline and we find smaller and smaller pieces, the coastline gets longer and longer. So there's a relationship between self-similarity and scaling. And let me show you quantitatively what that relationship is. Again, self-similarity says, and really I mean when I say self-similarity from now on, statistical self-similarity. Because that's what we have in biology. We really have exact self-similarity. Statistical self-similarity says that some property measured at scale A of R is proportional to, is a constant, times that property measured at scale of R. And scaling means that some property measured at scale R has a form which we'll see here as a power law, meaning that that property measured at R is some constant times R raised to a power. So this property here is the scaling property. So this equation is a small version, and this equation is a bigger version of that, but they're self-similar. Actually, in this case, they're just similar. But so to see that these two are related to each other, if I take this equation and plug it into this end of the equation and plug it into that end of the equation, which is what I've done here, if this parameter k equals a raised to the b, which is a constant, then I have the equation for self-similarity. So what this slide shows is that the scaling relationship leads to self-similarity, and therefore vice versa. So we have a two-way relationship between scaling and self-similarity. Now, this is the usual expression for the scaling relationship, but it turns out it's not the most general one. This is called actually the theory of homogeneous functions, and there's a more general form to this, and that's shown here, would have been shown in the first book that I wrote also. It's in my second book, but somehow just disappeared between two different versions of the book and just never appeared in the final pages. It just happened. This is the more complete form of self-similarity. This says that some property Q is a power law, R raised to some power, times a function. And this function is an oscillatory function, that if we add a certain value to that function, we recover the initial value. So that's all this says. And the function has these logarithms in it. And you can do the same thing here. You can show that starting with this, if we apply it to the both ends of the equation of self-similarity and we use the scaling thing, that we then recover the self-similarity relationship. So again, there's a relationship between self-similarity and scaling, and this scaling function also is consistent with self-similarity. In fact, this is the most general one. This is a little unclear because it's a messy-looking equation. I can show you that better in a graph. So the scaling relationship, the most common form of the scaling relationship, is that we have a power law form where the value measured at a certain property is constant times the scale raised to a power. If I plot this on a linear plot, I get a curve relationship like this. Actually, not exactly like this, but sort of close to this. But as you should have learned from, or as I tried to emphasize when I discussed plotting relationships earlier, this would be a pretty stupid thing to plot because we really couldn't tell the form of a curved line on a graph like this. So if I plot the logarithm of Q, of the measurement, versus the logarithm of the scale, I'll get a straight line on a plot like this. And therefore, straight lines on scaling plots like this are characteristic of fractals. And that's a phrase that's actually quoted in the article that Laura would have talked about today on the scaling in ion channel inactivation, that power laws are characteristic of fractals. So that's a phrase I like to use, and he quoted the exact phrase. So it's odd for me to see when I see my own words quoted. But especially when I have trouble of thinking what the phrase is and remember to saying it right. So, but I've done it here. So let me show you what that other more complex relationship looks like. And we'll even ignore this linear one because it's not useful to think about. This is actually not the most common form. This is the slightly less common form of the scaling relationship, where we have that the scaling is a power law times an oscillatory function. So here we have a straight line with an oscillation, constant period oscillation superimposed on it. And this is the more general kind of self-similarity. And as I said, this sort of self-similarity also comes out of the theory of homogeneous functions. So this is the more general kind, and actually the most general type of self-similarity. And let me give you some examples of self-similarity. This is from a physicist named L.F. Richardson. Richardson did a lot of really interesting things and a lot of odd things, some of whom the importance wasn't realized at the time he did it. Among other things, Richardson studied turbulence. When you have a river running down a lake and you have little whirls, and the whirls have whirls, and you have turbulence. And turbulence and the origin of turbulence is not fully understood. Richardson was also a Quaker and did not participate in World War II. He was drafted, I guess, and served as an ambulance driver. He was British in the front. And after the war, one of the interests he had in life was to try to understand the causes of wars. And as a prelude to do that, he thought maybe countries that share borders but have different amounts of natural resources are more likely to fight each other. And so he went to almanacs and was looking up the length of the borders between countries. And he found that they had different values in different almanacs, and he couldn't understand that. So as a precursor to his sociological study of the wealth of nations and their relative resources and populations, he wrote an article on analyzing the borders between countries. And this was an article that was published about 7 or 8 years after he died. It was published, so that's why it's called A Problem of Contiguity, an appendix to Statistics of Deadly Quarrels, what he was aiming at. And it's published in this odd place called the General Systems Yearbook. And what he found was, as he measured the lengths of coastlines or of boundaries between different countries, at finer and finer resolutions, the coastlines got longer and longer. And this is a plot of that. So this is a plot of the logarithm of the length of the coastlines measured against the logarithm of the resolution measured against the length of the coastline. And he did the resolution actually in two different ways in this article. One way that he did the measurement was he took a pair of dividers, which look like this, only they're made out of steel, and he sort of walked his way along the coastline. So he sort of found line segments that hit different points of the coastline. And you can see that if he used a finer line segment, the length would get longer because he would hit more of the pieces of the coastline. And he also used a map measurer and walked along the coastline. That's got a little wheel, and he walked with different coastlines of different resolution, which I've done also. This divider measurement worked a little bit better. And what he found, as is shown here, is that this represents the space between the 2 points of the divider, or the length of those little lines, and this represents the total sum of those lines. As he measured the coastline of Britain at finer and finer resolution, the length got bigger and bigger. In fact, the fact that this is roughly a straight line indicates that this has a power law scaling. And this was true for most of the things he looked at, for Australia and Germany and Britain and Portugal. For some odd reason, it wasn't true of South Africa. And he also did it for just a circle that he drew on a piece of paper. And this resolution bent over, but you can see that this is basically a constant line, that here this is a non-fractal. The resolution or the length does not depend on the resolution at which it's measured. Now, we see the same scaling not only in terms of coastlines, but in terms of the areas inside of cells. So Weibull, whose talk I once missed when he was in New York and shouldn't have, has done a lot of measurements of things within cells and has been using fractals to characterize the dimension of the cell membranes. So what he did is to look at resolution on electron micrographs and measure this as its length density, so it's really an area, because he's looking on edge on the membranes, of the inner and outer mitochondrial membrane and the amount of area in the endoplasmic reticulum. And what you can see here for all these three measurements is that as the resolution gets better as we go to finer scale, the amount of area measured increased. So there is no such thing as one value for the inner or outer mitochondrial membrane areas, or the rough ER. That the amount of area depends on the resolution at which we measure it. And this also shows another interesting thing about fractals and scaling, that very often we have a scaling regime which exists from here to there, but then it changes. In this case, all these curves bend over, because eventually we get to a resolution which is on the order of, in this case, atoms. And its size is smaller than that. Everything is resolved, and so we no longer have this fractal scaling. We have a single value that then characterizes the total area and is totally resolved. Now, an issue that's come up in a lot of different ways, and there was an article about this in Science recently, is if the scaling is only over a certain region, does that tell us, or is that useful to know, because it's not over every region, does knowing that something is fractal really help us if it's not fractal forever? And this rather hostile article in Science from David Avner, who should really know better, because he's done a lot of stuff on fractals, argued because fractals exist only 3 or 5 orders of magnitude, and he referenced as many articles as he could find about fractals to see what the range of scaling was for each of those. Therefore, he said the world isn't fractals, and all the stuff with fractals is a lot of crap, which is a very dangerous conclusion to reach, and not politically for people like me. But it was also not true, and this is the basic point. Having already talked to a few other people who study fractals, the point is not what the total range of this scaling is, but whether over this range the scaling tells us something interesting about how these things work as biological objects or how they're formed as physical objects. And in this case, the slopes of these curves are telling us as we go to finer resolution how many indentations these things have, and that may give us clues to their biological function, and it might tell us how they're formed as well too. So we can extract information from these scaling regions over ranges that maybe are 1, 2, 3, 4, 5 orders of magnitude, and even if the thing isn't a geometric fractal or a fractal forever, it may still give us useful information to characterize these objects, tell us how they work, or tell us how they're formed. And that's really the issue about them. Another example of scaling is the work I did in terms of not in space, but in terms of time, in terms of these ion channel kinetics. So in this plot, what was measured was the probability that a channel will switch between its open and closed state, given we measure it at this time resolution. And again, on this log-log plot, we have a straight line characteristic of a fractal, which is my phrase. And what this plot says is that as we go to shorter times, as we can measure the opening and closings of the channel finer and finer, the probability, that is how often it switches, is larger and larger. So the faster we can see, the faster the channel opens and closes. There's another way to interpret these plots, but I don't feel like doing that at the moment. The question is then, so what? In this case, the fractal scaling never bends over for this data, which you can see extends over 2.5 orders of magnitude. The limitations of the scaling regime are not that the scaling regime itself is limited, but limited by the range of times over which we can accumulate data. In this case, the shortest time by the resolution of the patch clamp electronics. In this case, at the long end, by how long we can hold on to a patch. And the question is, so we see this, so what? What does this tell us about what's going on inside the ion channel? And what we've been able to do is to interpret those scalings in terms of the physical structure or the motions inside the ion channel. And we've been able to show, as is true for other proteins, that if there's a large number of similar closed states that are not identical to each other, and a large number of similar but not identical to open states, separated by energy barriers, that this sort of structure would produce that fractal scaling. And that fractal scaling, the slope and shape of that line, gives us information about the energy barriers from one state to another. Or it may be that the energy barriers between states are changing in time. And in that case, the slope of that line on the scaling relationship and its form tells us how the energy barriers between the closed and open states are changing in time. In fact, for all of the channels we studied, as a channel sits in a state longer and longer, either the energy barrier to get out of that state stays constant, or the energy barrier to get out of that state increases in time. So it is as if channels become more and more stable in time and less likely per second to switch from one state to another. So what we're trying to do is to use these concepts from fractals to give us some better understanding of how these ion channel proteins are working. And also knowing that we have fractals, we can see how people have modeled other fractals and dealt with other systems, and that gives us clues in dealing with the system that we have as well. Other biological examples where scaling relationships have been measured, as we've seen, include the area of the endoplasmic reticulum and the inner mitochondrial membrane, the diameter of airways in the lung, the sizes of spaces between endothelial cells in the lung, and the surface areas of proteins. A protein is this big, complicated, folded thing, and as you get finer and finer, this big, complicated, folded thing, as you get finer and finer, there are more and more spaces, as I was describing in terms of the catalyst. And people have tried to use the fractals in terms of proteins to describe the area available to binding of ligands, as well as tried to show in some cases that receptors have different fractal properties, different fractal dimensions, than non-receptors, and it may be a way of identifying receptors or certain functionally important pieces of molecules. And again, emphasizing not only we have fractals in space, but in time, that we have the kinetics of ion channels, the opening and closings of ion channels. In chemical reactions, where the rates of the reaction are limited by diffusion, we have properties of fractals and in the washout kinetics of substances in the blood. Maybe what I'll do is I'll stop here, because I think where I'd like to pick up next time is maybe I'll describe a little bit about what's exactly going on in these chemical reactions limited by diffusion and in washout kinetics of the blood, and what it means for those systems to be fractal. So maybe this is a good point to stop, and we'll continue not next week, where we're all on spring break, whatever that means in Florida, and we'll continue in 2 weeks after I'm done flying back from Los Angeles. Okay, so that's a wrap.